Hello students, in previous part we have discussed about DNA replicative enzymes. In this part we will discuss on DNA replication. DNA replication. How the replication is copy of DNA can take place. This process is completed by the different ways, means simultaneously but different steps. All these steps are occurred at the same times. This is not we will refer with the part 1, part 2, but all these processes may be completed simultaneously. But to understand ourselves about the replication process, we will maintain the different part. During the DNA replication, the first step is completed. What one? Which one? Activated or activate the nucleotide. DNA nucleotides are monophosphate molecules, whether the adenine, guanine, cytosine and thymine. They are monophosphate molecule. Until they remain a monophosphate molecule, they are inactive. To activate these, there is requirement of phosphorylase enzyme. So first one is activation of the nucleotide. This process may be completed by the enzymes phosphorylase enzyme. How this enzyme activate the inactivate nucleotide? Inactivate nucleotides such like as AMP, adenosine monophosphate. It may be activated with the help of or in presence of phosphorylase enzyme. It change into ATP, adenosine triphosphate molecule. It means adenosine monophosphate. It contains only phosphate one molecule, and when it changes into triphosphate, it means phosphorylase enzyme makes two phosphorus molecules. Same as CMP, cytosine monophosphate. It also changes into CTP. ZMP, guanosine monophosphate. It also changes into ZTP. And thymine monophosphate. It also changes into TTP. This is the first step, which can be completed during the replication process. It means the first of all nucleotide which remains inactive, these are required to activate. To activate these inactive phosphate molecule, to activate with the help of phosphorylase enzyme. Second one is unwind the double helix structure of DNA. Unwind, or we can say that open the double helix structure of DNA. To unwind the double helix structure of DNA, there are following steps. First one, where the helicase enzyme may be attached with the DNA, because the DNA double helix structure is opened with the help of helicase enzyme. So we can say that in this process, helicase enzyme is required. But before open the double helix structure, we are identifying the site where the helicase enzyme is adjacent, and the site from where the replication starts, that is called ORI. So there are the following steps. First one is recognition of ORI. ORI stands for origin of replication, the site from where replication starts, that is called ORI. Replication site, that is called ORI. After recognize the ORI, we will attach helicase enzyme at that ORI. Second one is to attach the helicase enzyme कहाँ पे ORI. And this helicase enzyme unwind the double helix structure. The helicase enzyme unwind, or we can say that open the double helix. Double helix. And after that, we will attach topoisomerase, because topoisomerase enzyme remove the tensile force. It means whenever the two strings are attached with each other, twisted over each other, when we remove the unwind it, there is some tensile force which may attract to each other. So after that, topoisomerase enzyme. This enzyme remove the tensile or tensile force to remove the tension force, or you can say tension. And just after topoisomerase, we will also attach with the single strand stabilizing protein. Single strand stabilizing protein, so that both the strand may remain single. These are the following steps which we use during the unwind double helix structure. After unwind, but before unwind, we can see that there are two types of DNA structure. DNA is maybe linear or circular. If the DNA is linked, it may be such like this one. DNA is a double helix structure which has the polarity 3' to one strand, 3' to 5', and second one is 5' to 3'. First of all, we will recognize the ORI. The ORI, origin of replication. It means the site from where the replication starts. And we attach the helicase enzyme. This is the site. This is recognizing the linear. Second one is, and if we open it, we try to open it this one like. This structure is called replication form. Replication form. If the DNA is linear, it forms during opening the two strands, it forms replication form. If DNA is circular, and if DNA is circular, such like this one, DNA is a double helix structure which is circular. This ORI, if this side is the ORI, this formed a large bubble-like structure, and this is called replication bubble. So, if the DNA is circular, the ORI is replication bubble, as the name is known, and if the DNA is linear, then it is called replication. After this one, there is need to attach primer. It means the attachment or synthesis of primer. The third step is to synthesize the primer. What is primer? Primer is a DNA directed RNA polymer. We can say that for a primer is a DNA directed or DNA dependent RNA molecule. Name the enzyme which synthesizes the primer, that is primase. So we can say that with the help of primase enzyme, we synthesize the primer. And what is primer? Primer is a DNA dependent RNA molecule. As the primer is synthesized, we attach the primer at the just opposite to the 3' strand. After synthesizing of primer, it is always, or we can say it's always attached just opposite to three prime strand. Means polarity of 3 prime. Such like this one, if this strand is 3' to 5'. We attach the primer. What is primer? Primer is a DNA directed or DNA dependent RNA. Just like this one. The primer. One strand is 3' which contain OH group and other one is the 5'. This will also contain phosphoric acid. H3PO. We always attach primer at the just opposite to the 3' and 3' towards 5' strand. So this is the RNA or primer. It contains 5' here. This one is the 5'. 5' means phosphoric acid. And phosphoric acid will attach to the 3' contains OH group. OH, phosphoric attached to the OH, it forms the phosphodiester bond. So we always attach primer just opposite to the 3' strand or 3' polarity. After attachment, the synthesis or replication of DNA process starts. Next part is fourth one. Replication of DNA. In previous part, we have discussed about different types of replicating enzymes. Those all enzymes are helped in synthesis or replicating and replication. But one enzyme which not only helps synthesize or replicate the DNA, that is DNA polymerase 3. So replication of DNA can take place under the influence of DNA polymerase 3. How the replication process may be completed? Suppose this is the double stranded DNA. This contains 3' to 5' and 5' to 3. First of all, we will recognize the ORI. We know that just opposite primer is always attached just opposite to the 3'. And what is primer? Primer is a DNA dependent RNA molecule. We will attach the RNA primer from here. Suppose with the help of helicase enzyme, we unwind both the strands. With the help of helicase enzyme, after recognize the ORI, helicase enzyme is attached, and that helicase enzyme open the double helix structure. It contains 3', it will contain 5'. This one 5 to 3. Then primer is attached. Primer always attached towards the just opposite to the 3'. This is the primer. It contains 5th places and it contains 3rd places of R. As the primer is attached, DNA has the ability to make its copy. This strand is also has ability to make its copy, and this one is also. The copy will be formed after attachment of primary just next this one, like it means it forms nucleotide, and these nucleotides are attached from primer with phosphodiester bond and towards the nucleotide or 3'2 with the hydrogen bond. This one. This is the hydrogen bond. These are the nucleotide. This one is primer. As the primer attached towards just opposite to the 3 prime, nucleotide process will be formed. These nucleotides are attached with each other with the help of this is the phosphodiesterone. Phosphodiesterone. The bond which is formed between the nucleotide, that is called phosphodiester bond, and the bond between the nucleotide to the 3rd prime to 5 prime strand, that is called hydrogen bond. Same, this will occur, means the synthesis of copy of DNA can take place. In this strand, where is the primer is attached? Primer always attached opposite to the 3 prime. We have unwind this part only. So, if we calculate this part, 5 prime, this one is the 3rd strand or 3rd prime. So, the primer is attached here. The primer is attached here, it contains 3rd and it was the 5th. So, it starts to produce its nucleotide, this one. And it starts to form 3 prime to 5 prime, just like as 5 to 3. Sorry, not 5, it is 5 to 3. Because the primer is always attached just opposite the 3 prime. Is that clear? Suppose we open some more part of this DNA double helix. How is this one? If we open just more part, we have already started. Primer is attached here. This is the 3 prime to 5 prime and 3 prime to 5. The primer is attached here. It starts to form nucleotide. This one we have already discussed. Same as here. It contains. It starts to form nucleotide. From this one, this is the 5 prime to 3 prime, and this one is 5 prime to 3 prime. 3 to 5, so this is the part 3 to 5 here. Here is 3 prime to 5. This we have already discussed. But when we open some more part of DNA, then how the replication process may start? This process will remain continuous. There is no need to attach another primer because it remains continuous. It starts to form the nucleotide, this one line, till this one. But in this strand, when we calculate, when we try to replicate, we can see that this is the terminal end. So there is no synthesis of DNA particles. We attach one more primer just opposite to the 3 prime here. As we attach the primer here, it starts to form another nucleotide sequence. The forward. This one. Already, we always attach the number of molecules from this one and this one are equal. The 4 new synthesized here, then 4 new synthesized here. Here we need to attach one more primer. We can see that the template strand, which is 3 prime to 5 prime. This symbol can show that the template strand, maternal strand, and the new strand, which is synthesized, that can be always represented by 5 prime to 3 prime. This can be represented by arrow. It means this is the strand which is synthesized new, and this is the template strand. So always the replication process starts from 5 prime to 3 prime. Just if the template strand is 3 prime to 5. But in this one, we can see that the template strand is 5 prime to 3 prime. This is the 5 to 3. Then the replication is always from 5 to 3. Replication is always from 5 to 3. It means the replication, direction of replication is 5 to 3. But in this one, we can see that there are the primers present. If when we remove the primer, there will be some gap gap remains, and if the gap remains, we can say that this strand is not formed continuously. This may happen. For example, when you open both the strand, this process, this strand will be replicated continually. But here, one more primer is also required. As many primers are required, there may be some gap will be produced. And if gap produced, we can say that the strands are not produced continuously, and the strand which not continuously produced, that is known as discontinuous strand as well as lagging strand. After the fourth one, as both the strand replicate each other, suppose one more thing, if we stand both the strands separate, then there is no requirement of another, because all these strands will be synthesized continuously. This is 3 prime to 5 prime, and this one is 5 prime to 3 prime. But when we open this one, 5 prime to 3 prime, there is already required primer. After primer, we will separate some another design, because these are the nucleotides. Another one more primer is required. It synthesized nucleotide, and when we open it, one more nucleotide required. It also again synthesizes this one. So this is the discontinuous strand. This strand, if the template strand is 5 prime to 3 prime, the new synthesized strand will be always occurred 5 to 3, but it is discontinuous. When can we see that discontinuous? Where we remove the primer. The next step is removal of primer. Removal of primer. So step number 5. Primer is removed with the help of DNA polymerase 1. The enzyme DNA polymerase 1 enzyme is required. Suppose this is the 3rd prime to 5 prime, and this one is 5 prime to 3. 5 prime to 3. As we have discussed in the last session, this is the primer contained, and here is primer, here one more primer, one more primer. But in this step, continuous strand is produced, because if the primer strand, there is no limit, no terminal end. Until the terminal end, nucleotide will be continuously synthesized. But in the first session, we have seen that this one, ours, the nucleotide will be synthesized this structure of this one line. It is terminal end. One more nucleotide is required, a primer is required. As we synthesize, as we open the double helix structure. And here one more primer is required. As we remove the primer is removed by DNA polymerase 1. As we remove the primer, some gap will be shown this one. Primer is removed by DNA polymerase 1. There are some gap will be present. This gap is filled up with DNA polymerase as well as ligase enzyme, because it forms fragments of DNA, and fragments of DNA are attached with each other with the help of ligase enzyme. So we can say that here is ligase enzyme required to fill the gap with RNA polymerase. So in this strand we can see that this strand is continuously producing. So it's called continuous strand. But here we can say there are some gaps present, and these gaps are filled with the help of DNA polymerase 1 and a ligase enzyme. So we can say that this strand is discontinuous strand. This is the first major difference. Second one is the strand which continuously produce that may be produced earlier. It means it takes short period of time. So it is also called leading strand. Why it is called leading strand? Because the strand 5 prime to 3 prime continuously, just opposite 3 to 5, is continuously produced, and it produced earlier than this one. So it is called leading strand. Whereas the discontinuous strand will take some more time rather than this one. So it is called lagging strand. As well as it also known as Okazaki strand. Why it is called Okazaki? Because we have seen that there are some gaps present here, and gap present strand is known as Okazaki strand. Why it is called lagging strand? We have seen that. Why it is called lagging strand? Lagging strand is a discontinuous which takes more time than the leading strand. So it is called lagging strand. After formation of these strands, there is another enzyme required to proofreading. Is the replication may be completed same sequences or some other sequence. Some mutation cannot take place. Then the step number 6, proofreading. And the enzyme which is used in proofreading is, first one is the unit of DNA polymerase third, epsilon. And nucleases. Both are the enzymes which are required to proofreading. It means the synthesis of new strand is completed to just like as this template strand or not. If there is mutation can take place, they firstly proofreading and then repair. So there is no mutation can take place. The proofreading and repairing work is done by the help of epsilon. Epsilon is the unit of DNA polymerase third. Because we have seen that DNA polymerase have 3 units: alpha, epsilon and beta. This epsilon unit help in proofreading and nucleases in help in proofreading as well as in repairing. So there is no any types of mutation can take place. Clear? This is the replication process. Now, there are some scientists which help to synthesize the DNA molecule with the help of IVF, in vitro fertilization. There are 3 scientists. One from India, Hargobind Khurana. Hargobind Khurana, the Indian scientist who synthesized DNA in IVF without template. Means there is no requirement of template strand. Without template strand, he synthesized DNA. After that, after his discovery, he was awarded with the Nobel Prize. Means there are 3 scientists. What is the role of synthesis of DNA and RNA? There are 3 scientists. One is Hargobind Khurana. Khurana, not Khurana. He synthesized DNA. DNA in IVF, in vitro fertilization without template. It means there is no requirement of template strand. And second one is Kornberg. He also synthesized DNA in IVF, but it took with the help of template. Kornberg is also synthesized DNA in IVF with the template. Hargobind Khurana synthesized DNA in IVF without a template. One more, Oka. He synthesized RNA in IVF. He synthesized in RNA by IVF in vitro fertilization. IVF is a test tube. So after this discovery, Har Gobind Khurana was awarded with a Nobel Prize for his discovery of synthesis of DNA without a template. This process is also known as semi-conservative. In next part we will discuss on how the semi-conservative process can take place. Clear?